I'm Danica Helb. I'm a PhD student in infectious disease and immunity at the School of Public Health here at Berkeley, and I'm very pleased to introduce you to Dr. Bernhard Weigl. He is the director of the Center for Point-of-Care Diagnostics for Global Health at PATH. And Bernhard is the inventor listed on over 65 patents and patent applications, and has over 70 publications in his name. He received his PhD in analytical chemistry in 1993 from Graz University. And in 1996, he was a scientific co-founder of Micronics. In 2003, he co-founded Microplumbers Microsciences. And in 2004, he started as the Diagnostics group leader over at PATH. So I'd like to tell you a bit about PATH. PATH is an international nonprofit organization that works to develop sustainable and culturally relevant solutions to a number of global health issues. Some of these issues include emerging and epidemic diseases such as HIV, malaria, and tuberculosis, maternal and child health, ensuring safe childbirth and trying to eliminate diarrheal diseases in children, health equality for women, including family planning, vaccine development and dissemination, and health technologies, including clean water and also global health diagnostics. So at the Center for Point-of-Care Diagnostics for Global Health, basically this group integrates a number of key components to developing a successful diagnostic test. So first, they work with the community where the test will be implemented to assess the needs for different diagnostics. They also work on research and development, both in labs at PATH and with collaborating institutions to develop these new products. They validate these tests both in a laboratory setting and in a field setting. They work to help commercialize these assays. And they're also very involved in training, both training academic leaders to develop more relevant diagnostic products, and also training the end users so that these products are used properly. And there's a number of different diagnostic technologies that are going on in the laboratory at PATH. So Bernhard will tell you more about these, but basically he's working on a bunch of immunodiagnostic platforms, molecular diagnostic platforms, and new platforms that either use isothermal amplification or non-instrumented amplification techniques. And so I'm pleased to announce Bernhard, please.
All right. Okay. Well, thanks so much. Thanks for inviting me here. And it was a pleasure. It's not here. Yeah, I do. I do have my cell phone here. No? Do I? Yes. So it was wonderful to see Paul's presentation before. Of course, brings up all the memories of things we did together. And it's also, there's another reason here. And I really think when we first started out with microfluidics, global health was really not on the forefront of anybody's mind. I mean, this was, the idea was to come up with diagnostics that would be useful primarily for developed countries. Cost was an issue, as it always is in healthcare, but it wasn't probably the primary driving force here. But in the last, I would say, 20 years now, 20 years, global health has become an extremely attractive new research area. Especially in the Seattle area, as you all know, we have several institutions that focus on that, with the Gates Foundation driving much of the research, and then at the university and at PATH and some other institutions as well. And also, the fact that this conference exists, that this group here exists, is a testament to the great interest that especially students have in global health. And in fact, we see it every year when we put out job notices or training courses or things like that. There's lots and lots of people who are interested in this. We are in the great position of being able to pick among the very best people that we can get here, and they are even willing to do this without stock options, without huge monetary rewards, because it's a very intriguing area to work in. So the other thing that Paul's talk this morning sort of brought back for me was when I looked at these technologies and looked at this work that his group is doing right now, the first thing I was thinking was, I want to do this. This is really, ultimately, I'm a technical person, so this is the coolest thing. However, I'm working at PATH now, and at PATH my role is, while we still do a lot of technology development, my role is slightly removed from that, and that's represented, I think, also in this talk today. I'll take a step back from immediate technology development and talk about sort of the context for that, the context for diagnostics for global health, also some of the things. So first I want to talk about global health and the role of diagnostics within global health. A little bit about the transformational potential that point-of-care diagnostics can play in low-resource settings. Bit about platform development at PATH. Then something I think that when people think of global health, they don't really think about this, at least not yet, and about chronic diseases in low-resource settings. Very critical problem, huge area, just emerging as an area of interest for most people. That is not yet reflected in research, but needs to be. Another very important part is sustainable commercialization. I'm going to talk a little bit about technology transfer and manufacturing models for and in developing countries. Paul has sort of alluded a little bit to the idea of low-cost manufacturing, and I'm going to build on that a bit. So first of all, and this is true not just in developing countries, but also in developed countries, or this really number is from developed countries. Diagnostics comprise 3 to 5% of healthcare spending, but influence 60 to 70% of healthcare decisions. So essentially, we as diagnostics developers, manufacturers, can always argue that diagnostics is a very good deal. You get a lot of information, you get a lot of value out of doing diagnostic testing. Another thing, diagnostics in general is, while people understand the need for it, it seems to be sort of the ugly stepchild of medical care systems. People think of medicine as, there's a drug. The drug is sort of the silver bullet. You give a drug when somebody's sick. You give a drug, and that solves the problem. Right? Or in some cases, even now in global health, we see this with vaccines. So if you have a vaccine, you don't have to worry about anything else. To some degree, that's true, but to a large degree, it's not. So I'm presenting this slide here as what I think, actually, when you think about a whole healthcare system, how do you want this to work? And I think information is really at the core of any healthcare system, at least should be. If things were to function rationally, where there wouldn't be any entrenched interests, any preconceived notions, existing medical specialties and so on, how would you go about designing something like this and prioritizing healthcare? Well, the first thing you probably want to do is you want to know what diseases you have in a population. So that's epidemiology. That includes things measuring, and all these things have measurements at their core, because everything else, otherwise it's just guesswork. So you have to measure things, and that's what diagnostics ultimately do. We measure parameters. And I use diagnostics here in its broader sense of, surveillance tools, quality control tools, and so on, every time you measure biochemical parameters. So you measure levels of diseases, levels of toxins, both in patients as well as in environment, pathogens that are circulating, other kinds of diseases. This is not limited to infectious diseases, of course. So you have all these things. You then have to do a certain prioritization steps, because there's always way more potential threats and potential diseases and real ones than you can actually deal with. You can only deal with the ones that have most impact. So based on those parameters that you measure, and you determine the impact on morbidity and mortality and also projected future risks, sort of emergence of new diseases, emergence of problems, behavioral issues, you can prioritize what diseases one would want to address primarily, and obviously you prioritize this by funding. Then diagnostics, the classical diagnostics is individual patient diagnostic testing. So using a combination of clinical, laboratory, and patient feedback methodologies, you determine what a person actually has. And it's perfectly valid to use epidemiological and syndromic information as part of that diagnosis. I mean, it's not just measuring a biochemical parameter, because if you know that a disease is circulating or is not circulating, then you don't have to measure that. So it's reasonable to not just rely solely on biochemical or microbial parameters. And then there's treatment and prevention. That's drugs, therapy. That's really where most of the money is spent right now. So the vast majority is really in only this one area here. Once you do the treatment, you have to determine, is this treatment effective? And in developing countries, that's particularly hard because it's hard to have, you have a lot of loss to follow up. Infrastructurally, a lot of the times you're not able to get the same people back for follow-up visits and so on. So this is something that is difficult to address in developing countries. Verification of cure would be sort of beyond the individual treatment. You then measure, is this really gone? Is this disease gone? And so on. And then with all this cycles back at some point. So you introduce a new intervention, then you do another population-based surveillance study and see if overall that disease has been reduced or the impact of that disease is either at least managed or ideally is decreasing. That's really what it's all about. So you keep doing it, you keep iterating on that. There's a few other things you could do with tests, and you should, and these are in the areas of QA, QC of diagnostics. So you need to really test the tests themselves and test the drugs, test the performance both as you're manufacturing them and then as you use them, and then you measure the impact of certain interventions again using, in many cases, biochemical measurements. Same for drugs. You need to make sure that the drugs that you're giving or the vaccines and so on are actually working. So we heard before what PATH does. We have a fairly holistic view of healthcare in global health, and we try to do it in a rational way. So I think that's really one of the things that maybe sets us a bit apart from many other nonprofits that work in developing countries. We look at a fairly big picture. We are a relatively large organization. We have about 900 people worldwide, and we think about anything that we introduce really in the context of a whole healthcare system. Some of these factors down here, you probably know that a significant chunk of our funding, more than half, comes from the Gates Foundation. My group is about half, and then the rest comes from the US government and other foundations as well. So what do we do in product development? Products for developing countries, generally, if market forces were sufficient to create products for developing countries, they will be there. They aren't for the most part. So there's a need for something that's generally called product development partnerships. I'm sure we hear about this afternoon, probably from Cepheid as well. These products generally, it's difficult to create them purely based on sort of return on investment kinds of strategies. So what PATH does is we do certain activities that reduce the development risk and because of that increase the potential return so that a particular product still falls into the general comfort zone of private enterprises. So this thing that we can help do, and we do this of course with external funding, is essentially it helps the public benefit. So we work on a bunch of different technologies, vaccines, diagnostics, and over-the-counter products ranging from sort of command and control kinds of interventions like vaccines to more like provider-directed interventions like typical diagnostics and consumer-directed interventions such as buying clean water or water purification, household purification systems and such things. There's lots of projects that we have going on at Path Diagnostics at this point. It's a bit misleading because all of us have quite a few projects going on at any given time. But still, you can sort of get a basic idea of sort of projects ranging from infectious diseases to a little bit now in chronic diseases, ranging from immunoassays to molecular assays, and then sort of technology-neutral projects as well. Point-of-care testing in developed countries. So there's, I think, a fundamental difference in how point-of-care testing, or kind of role point-of-care testing can play in developed and in developing countries. In developed countries, diagnostics, point-of-care testing is defined as diagnostic testing that is performed near or at the site of the patient care, with the result leading to a possible change in the care of the patient. Well, that's the uniform across. But the key objective is to generate a result quickly to influence treatment. So that's really what it's about. It's not necessarily about cost. It's not necessarily about sort of enabling certain things. In some cases it is, but it's mostly, can you make better decisions because you have the information more quickly? That is not necessarily, that is part of the driving force in developing countries, but it's not the only one. Point of care here in this country and in Europe to a lesser degree is designed as an adjunct test to central lab testing, not as a replacement. Here the assumption is you'll always have a central lab, you'll always follow up with additional testing, but point of care is sort of the first line of defense that you have. You do it right away, you get a quick and dirty piece of information, you act on it, but then you follow up. That's kind of the model. I think because of that model, or maybe because of other reasons such as entrenched interests to some degree, but I think also because of that model, point of care has not really been a transformational technology. Point of care is important, it's grown, there's lots of companies working in this area, but if you look at diagnostics overall, vast majority is still lab-based. And sort of every new device that comes on board, it's kind of cool and important, but it's not like it's changing anybody's life, with the exception of diabetes. Diabetes has been point-of-care testing or home testing has been a huge life changer for diabetics, of course, over the last 20 years. For other diseases, we have not seen that yet. We may yet see it. There's initiatives in that direction, but so far we haven't. Point-of-care testing in developing countries is a completely different ballgame because point-of-care testing in some cases is really the only thing that's there. Because in many cases, there aren't any central labs, or whatever there are, they tend to be reference or training labs, but they would not handle the bulk of the diagnostic volume that has to happen. So here is just some examples of where point-of-care testing might be used. Sort of up here, you can see the sort of the marketplace setting up here, where somebody's doing it essentially as a sort of a little commercial franchise to do some testing. Here is a blood screening campaign that's very well organized, but it's still in open air. This is sort of a very typical kind of sort of mid-size hospital clinic setting where you may well have a few hundred patients come through every day. But that sort of would be the lab area, this and maybe another desk or something like that. That's kind of the extent of what you have to work with. The lab, I mean, these are, I'm showing pictures. These are maybe particularly rough ones because they're sort of instructive. And of course, there are beautiful labs in developing countries as well. So don't get me wrong. I've seen labs that look just as good as the ones we have in our lab. But you do see these kinds of things. You see things that are, they're not climate controlled. They're not, they're not dust-free. The water wouldn't be purified. No refrigeration. No continuous power. In almost all settings, you probably have some kind of power, but it might die after 3 or 4 hours, and then so you couldn't run assays for a long time. You might need something like this, a battery. Transportation wouldn't be in a refrigerated truck, but maybe in something like this, so it would be exposed to high heat. And really this is, if you look at labs, what do they actually do? Well, what they actually do is run strip tests. They have microscopes, they have strip tests. That's the vast majority of diagnostics labs in developing countries, that's what you have. So what is the sort of the transformational opportunity here? I think there's both a challenge. There's no existing, or there's, in many cases, there's not a lot of existing point-of-care infrastructures in developing countries. At the same time, that's an advantage because you can really design, a diagnostics infrastructure to some degree from scratch. At least it's a possibility. It's something that point-of-care testing might enable. The analogy might be the wireless phone versus landlines. Many places you probably where you don't have any communication infrastructure, you probably don't want to install landlines, you install wireless phone lines right away. So you kind of do a leapfrogging of traditional lab-style infrastructure. But the question still remains, will central labs be a niche? Or could they be a niche or an adjunct system sort of as a reference lab rather than something that actually does the bulk of the testing? So there's some good rationales for why this might play out that way. The flip side of that is biosamples are not the same as electrons. I mean, you can scale up a wireless phone network fairly easily because those phones simply, they don't really care if they run at 50 degrees Celsius. They work probably reasonably well there. You don't have to refrigerate them. They last, they're in many ways they're much simpler devices than diagnostic devices. So it's really not the same thing. And the other thing is QA, QC. Can you really do this in a completely decentralized setting? You have to build a lot of intelligence into your devices or into your instruments and to some degree into your training system and validation system to make that work. Because that's really one of the big drivers for central lab infrastructure in this country is quality, the ability to control the quality. So challenges are higher per test cost in most cases, quality control risk, ability or availability of trained health workers in village settings. Education generally leads to people leaving the village setting and going to places where they can make more money. So that may be difficult. Procurement and distribution, you still have to get the tests. If you either have to get the patients to the capital, which is hard, or you have to get the tests to the villages, which is hard. So either way, it's hard. We think telemedicine can be a supplement in a sense that Paul talked about it. The cell phone can measure and transmit information from a test. But to some degree, it can also disempower local capacity because if you have somebody else make the decisions based on something, so if the local person only takes a picture, it gets sent to some central place and then the directive comes back, that might be useful. But sometimes we think, well, maybe it's better to actually train the local people in the meaning of the results and also in the interventions, because then you create some local capacity, which is then helpful for other things as well. Sometimes central lab is really the only solution because there's no point-of-care test available or even foreseeable. Advantages for point-of-care testing is in principle lower startup cost because the infrastructure requirements are lower. You can scale it. You do as many as you can afford. You have more flexibility. You can customize devices for different settings. You can have in some settings with low throughput, you have one kind of device. Medium throughput, you have a different kind of device. Higher throughput, you have yet another device. That's something that's in principle much more cost effective. It does empower local healthcare because somebody locally can actually do something about a disease, can get information and do something about it, which is critical. Rapid turnaround can be critical, especially for acute diseases. And sometimes point of care is really the only solution in cases where you have to have a quick answer, because otherwise you need to determine treatment immediately, as opposed to 3-4 days later when the patient might be dead or the disease might have run its course. So when we think about diagnostics development, we think about profitable diagnostics are the ones that ultimately are most likely to be available over the long term. But market forces alone are not sufficient to ensure diagnostics availability. We also strongly think that if you have an expert panel and design diagnostics based on expert panel's opinions, you will not come up with a good diagnostic. You will have to ask the users. You will have to do what's called user needs assessments, what we call that. And it's an involved and complex process, but I think it really leads to better diagnostics. We also don't think there's a silver bullet, where we frequently disagree with the Gates Foundation because we think that there's usually not just one way to do things, and there shouldn't be just one way to do things. In the Gates Foundation, frequently there's this tendency of sort of trying to find the solution, the ultimate solution to a problem. And in reality, that's just not how science and development in general works. I mean, there's improvements, there's sort of in-between steps that I think are very valuable that are necessary. So we remove risks for all partners, technical, market, clinical, and uptake risks. We work in all these areas with ideally resulting in viable diagnostics products with maybe somewhat lower profit margins, but still viable profit margins. Here's an example. That's something we're almost done with right now, this project. We're developing a little strip test, standard strip test for Chagas disease. Chagas is a regional disease. It's a neglected disease. You have only about 100,000 new cases every year. So the market is really not quite big enough for any major manufacturer to sort of get into the game here. There are some pretty good drugs, but they are kind of nasty. They have significant side effects. They're essentially like chemotherapy agents. And so you don't want to give those drugs just on syndromic information. So you do want to measure if you have Chagas or not. A strip test, right now there's ELISAs that work pretty well, but the people who have Chagas disease are the ones that live usually at the edge of the jungle, edge of the forest. It's a very rural disease. So in the city, you won't find it very much. So obviously, those people, it might take them easily 2-3 days to get to a place where they could get tested, which is a limiting factor. So a strip test can help with that. There's nothing particularly innovative about making this strip test. The reagents are all out there. But what PATH brought to the table here is the knowledge how to make strips, the knowledge how to find the right manufacturers, find the right distribution partners, and put together essentially a model that will ultimately result in, we think, a sort of a sub-$2 kind of assay. That's a strip test, and we have this currently working. We have done some clinical validations already, and we expect this to be available in 2011. Paul already mentioned lateral flow strips, and they are, in some sense, the reason I'm mentioning them because that's really what PATH has built its reputation on originally in terms of diagnostics. Lateral flow strips are, in some sense, the perfect diagnostic in terms of ratio of complexity to result. They are incredibly simple, have very few parts, no moving parts. They do sample preparation, sample fluid movement, signal visualization. They have an internal control. They contain the bio-waste. And they cost, there's lots and lots of manufacturers that can make those and sell those for less than $1. So that's really, there's no other diagnostic that can do this at this point. In fact, the cost of goods for these kinds of things are now approaching typically about 18 to 20 cents. So you really have even a reasonable profit margin on this. There's downsides, of course. Right now, they don't have necessarily sensitivity, specificity, the ability to detect active infection, and also only limited ability to do quantitative measurements. So, of course, molecular tests are still needed. But when we say that, so we do molecular diagnostics at PATH. But what we do is every time we think about molecular diagnostics or we try new approaches, we think about, could we do this in a way that is similar to how a strip test works? And we don't mean necessarily technically, but functionally. So is there a way to do away with the pump, the plastic, the reader, the heater, things like that? All the kinds of things that a strip doesn't need, the fridge, can you dry everything down? Can you do things functionally similar to a strip test? And so in terms of amplification chemistry, we have focused our area after also doing a lot of work with the traditional microfluidics originally, the 1.N versions. But now we've really focused on isothermal amplifications because they have some significant advantages over PCR, especially in that they can use somewhat dirtier DNA, or at least they have a different set of inhibitors, maybe a less significant set of inhibitors than PCR. So less sample prep is needed. In some cases, you can, for example, take a dried blood spot on the filter paper, cut it up, throw it into the amplification vial without any sample prep at all. And in some cases, you can do simple sample prep. Isothermal amplification has inherent advantages because it's isothermal, meaning you don't have to cycle, you just heat. In some cases, isothermal amplifications run at relatively low temperature, which means you don't have to heat much. And some of them are not very sensitive to the particular temperature, which means you don't have to heat very accurately. So these are important things that help with simple diagnostics. Materials, of course, are critical. So we're looking at, we have to make sure that both the materials that will be used in these devices as well as the manufacturing processes to make them have to be low cost and field friendly. And we're thinking about these kinds of diagnostics as interchangeable modules. So we took a step back from completely integrating assays, like we originally tried to do this with Micronics, to something where we say, no, users can do some steps, not many, 1, 2, 3 steps maybe, but it's okay maybe to transfer a liquid or to open a cap and put something else in. So we think because of that, when you do that, you allow yourself, you can make your assays significantly cheaper. So here are some examples of what we do here. The sample prep, we came up with a few different methodologies. For amplification, we're working with things like LAMP and another one called recombinase proteinase amplification, and then a third one with the Keck Graduate Institute, XPAR. These are all very interesting methods. The good news also with isothermal methods is there will never again be sort of the kind of stranglehold that Roche had for the longest time on PCR. Will never again happen with isothermal methods because there's now about 15 validated methods out there for isothermal methods. Some of them not as good as others, but there's at least 3, 4, 5 different ones that are quite good. And so even though companies are positioning themselves and acquiring some of these technologies, I don't think there's going to be one that's going to have the one technology. We also came up with a way to replace heating. I'm going to mention that in another slide. And we do some work on signal detection as well. So sample preparation, the thing on the right here is sort of the standard approach to sample preparation, the QIAcube. Of course, you can get a somewhat simpler device, but still, it's a complex, automated, more or less complex device. The same kind of sample prep you could do with the kinds of things that I have up on the top left, which is somewhat manual. You have to do things with a syringe, but if you do that, you have to train people to do that. And yes, you can make mistakes, which was one of the big drivers to move away from that sort of model. But at the same time, what are you trading in? So you're trading in something like, and it's a reasonable argument that's being made at this point. There's devices such as the Gene Expert, for example, that I'm sure we'll hear more about today, that are integrating all functions. The Macronix device is doing the same thing. Very exciting in principle from the perspective of a low resource setting user, where you don't have to have a lot of training. You really can put in a sample, you press a button, you get a result out. But what you have to pay for that. It costs many thousands of dollars, and probably tens of dollars, or at least $10 for the disposable. This thing here has cost of goods of about, certainly less than $1 for the sample prep. But you have to do some manual work. So can you train? Can you train this? Can you do this in a practical way, in a reproducible, safe, high-quality way? Usability studies have to show that. But we think that there are some settings where this is the right approach. There's other settings where probably something like the instrument-based approaches are the right settings. Same for there's another sample prep method that we came up with down here. That's a card-based approach, similar to the dried blood spot method, but it already filters out the red cells, so it does one additional step. Heating, we came up with something that heats. So we use exothermic chemicals for heating, essentially hand warmers or chemicals like that, and we combine that with something called a phase change material. That is essentially anything where you have a solid in equilibrium with a liquid. And ice water is an example. Ice water always has 0 degrees Celsius. You can have wax mixtures, other kinds of mixtures that have very, very clear set points, temperature set points, very low cost, dirt cheap reagents. And so, for example, we came up with methods to do to heat for the 3 isothermal methods that I talked about before. Recombinase proteinase amplification needs about 37 degrees Celsius. The LAMP needs about 60. XPAR needs about 55. And so we came up with formulations for those 3, and we have those. We had studies done with those already. And so that's really a way to get rid of the heating portion of the experiment, which really was in some ways the only remaining instrument component for isothermal amplification methods. And so this is what this looks like here. Essentially, it's a little plastic thermos, costs about $4, and it's actually reusable. So the thermos itself is reusable. All you replace is there's a tablet that contains the heating material. You put it in. It's equivalent to a reagent replacement. You put it in. You add water. You can use any kind of water, really. And you put your centrifuge tubes in, close the lid. It starts. And right now, we just we time it. So you have to take it out. But we also have dyes that are cumulative heat indicators that can actually tell you by color change when you're done with the reaction. So this is a very simple way to do isothermal amplification. Visual detection, this is not necessarily our inventions, but this is just the various ways how you can do this. There's LAMP uses, for example, allows you to do turbidity-based or fluorescence-based detection. Fluorescence usually makes you think of an instrument, but really you can do this with a keychain LED. You can visually look at these things. There's also a dye now for LAMP, and there's another one in development for RPA that gives you visually an absorption-based color change reaction that tells you presence or absence of a pathogen. And the other alternative is to use a strip test, again, similar to the strip tests that I talked about before, but these strip tests are detecting amplicons as opposed to antigens or antibodies. And those work wonderfully. In fact, the strip tests right now, they give you single copy sensitivity with our assays. So they're just as good as PCR. There's really no difference. There is, however, one significant thing that a lot of people are trying to address right now, and that is containment of amplicons. You don't want your amplicons to kind of end up in your work area and mess up everything else. So containment is very critical. We think it's a solvable problem. but it's actually something you really have to think about. How do you do this? And how do you do this in a very low-cost way? There's some things you can buy now, some plastic containment devices. We've tried one where we just put little Ziploc bags two into each other and work inside the Ziploc bag. That actually works quite well and costs about 10 cents. But it is something where you have to rely a little bit more on the user to know the assay a little bit better. So it sort of limits the settings you can go. Chronic diseases I mentioned as an area that is emerging, and it's absolutely critical. I don't know, yeah, I have this here. I wanted to show you this one first. So diagnostics, I said before, comprise 3.5% of healthcare spending, but influence 60 to 70% of healthcare decisions. Chronic diseases comprise more than 50% of morbidity and mortality in low-resource settings. In some cases, way more than 50%, but receive only 2% of the total R&D funding at this point. So right now, if you try to do a chronic disease for low-resource setting projects, you'll find some private foundations and a little bit of NIH money to go after. But it's huge. I mean, everybody thinks about things like HIV, malaria, and as important as those are, HIV affects way fewer people than diabetes, for example. And diabetes is a deadly disease in many places. This is not a sort of a nuisance kind of thing. These things, when we talk about that, we mean, cardiovascular diabetes, which is really one disease for all practical purposes, and cancer, which is sort of a little bit separate because it needs to be addressed very differently from cardiovascular and diabetes in terms of diagnostics. So we are focusing right now on this cardiovascular diabetes complex. And right now, if you had, let's say, $50 million and you wanted to spend them on one thing that would make the most difference in the world in terms of diseases, you would want to address diabetes, and you want to somehow figure out a way to reduce cost and increase access of diabetes screening and treatment in India and China. That's the single biggest thing you can do with your investment that has the single most impact in the world. Most people don't know that or understand, or even the Gates Foundation really does not at this point address chronic diseases in any significant way. New technical, clinical, and business models are needed for chronic diseases because this is not a one-shot deal. Chronic diseases are chronic. It means you have them for a long time. You need to do something for a long time. There's not one vaccine, one drug that solves the problem. You cannot really eradicate them. You have to manage them, which means you need to figure out ways to make that kind of treatment and access to that kind of treatment simple, low cost, and practical for developing countries. I don't know if I have this. I mean, one of the things I just wanted to bring up here, because ultimately, if you think about diabetes, what you have to do is you have to screen. But let me go through the other slide first. So we're working on sort of two channel areas right now for diabetes screening. And one of the things, of course, that always comes up is, so once you know if somebody has diabetes, what can you do? And the thing is, right now, treatment, classical Western-style treatment, is largely unaffordable in many settings. But you have to look at incremental improvements here. So you have to do things like, for example, diabetes is not something you can conquer completely, but you can extend lives, you can reduce impact, you can reduce cost of the disease to society. So even if you can't maybe afford full-scale treatment, you might be able to afford lifestyle programs, some advisory projects, things, better primary health care, things like that has an impact on diabetes. And even if it only sort of averages, a 5-year extension of life or reduction of cost of associated diseases and so on, that would be very significant. But here are some screening approaches first. So fasting glucose is impractical. That's the standard method here, is impractical because in most healthcare visits are, I mean, I say random, but what I mean is they're driven by something else. So somebody comes in because they have some kind of condition that needs to be addressed. And so the only time you really can do glucose screening in most cases is if you get people in randomly, and you can't, they wouldn't come in fasting usually. So you need to use methods that don't require fasting. There are some around, like HbA1c, that's glycated hemoglobin measurement. There's a very new, interesting area that is called advanced glycation end products that both really rely on the same idea. Means glucose, if you have too much glucose in your body, it starts binding to various things in your body, including hemoglobin, including skin, various tissues. HbA1c requires you to take some blood, but you can do some simple testing on that blood and measure HbA1c. The advanced glycation end products actually work through non-invasively, just with autofluorescence. And what we're working on right now is to see if any of those methods are sort of practical and how they would have to be modified to become practical as primary screening tools in developing countries. So very quickly now, the last thing here is I wanted to address commercialization of diagnostics. So it's critical, a commercial focus on low resource settings we think is critical to get any company to make a product that makes sense in developing countries. So we have found that while it is, in some cases it works to have a Western or a large diagnostic company take the lead in developing a product for developing countries, and we do this, we have done this too with QIAGEN, for example. Cepheid is another example. So these things do exist, and it does make sense in some cases, but more often than not, these kinds of projects are seen as sort of altruistic kinds of projects that simply do not get the support from the board, the support from investors that are needed. So we find that transferring technologies and manufacturing in low resource settings provides the commercial focus, provides the capacity building, provides the right cost structure. And yes, there's support needed in developing countries, for companies in developing countries in training, QC, QA, even business functions, but we think there's a lot of value. We have examples here. We have transferred lots of tests to India, for example, Indian companies, and some of the outside of diabetes care, some of the biggest sellers of diagnostics of any kind are actually the ones that are now produced in India. The malaria strip test that came originally from Path, that is now manufactured by Orchid, they make about 100 million every year now. And so that's, these are sort of large numbers. And the reason this works is because their whole market is in developing countries. With them, we don't have to come up with complex contractual arrangements that require them to sell at a certain price, because they sell at 60 cents per test because they know that's the market. It's not, they don't have to, we don't have to force them to do that. We do the same, we've announced in a new initiative in Africa as well with Camry, where we are trying to start a new production facility in Kenya that's a parastatal, and we hope that they can also do strip test manufacturing and hopefully eventually things like the microfluidics, the paper microfluidics, because we feel that the same kinds of technologies that are needed for strip test manufacturing are also, or similar level of technologies would be possible for paper microfluidics. So they could do the same kind of thing. And yeah, I'm going to leave you with this slide here, just to show you this is not all theoretical. These are real products, have changed the lives of lots and lots of real people. And I hope we can do that again. Thank you.
Thanks. We're a bit behind schedule, so we only have time for a few quick questions.
Hello. Yes, my name is Madhu Varma from Intel Corporation. So I have two comments. I guess one of them, you made the comment that for most part there is no real POC type product except for diabetes, which is a true statement. But I wanted your sense whether it has a lot to do with, especially in chronic diseases, to the fact that there are no simple markers known for complex diseases, or is it just a pure function of technology, which in infectious disease, especially for molecular or nucleic acid, yes, that's true. But what about, even for diabetes type 1, what would, any comments there?
I mean, there are certainly some diagnostics that would be difficult to imagine doing on the point of care. But I would say if you ask the technology developers here, I would say the majority of the kinds of things that you would typically do in a central lab testing setting, you could in principle do in point of care. It's a matter of cost, engineering, but it's not of overwhelming complexity.
Okay. The other comment I wanted to make, that it might be useful to talk later, Intel has one technology it's not directly transferable today, but I think it has the potential to go to the kind of cost structures we are talking on the protein side. So I'd like to talk to you.
Very
Thanks.
interested.
Anyway, my question is, what has PATH done in terms of addressing quality control in the field? I mean, it's serious. You're way out there. Other places are making these strips or devices. What do you do for some kind of centralized quality control to ensure accuracy to a certain degree?
This is the big problem. This is we have had some success with reporting requirements. So we have licensing schemes that require some companies to report back to us and even send batches. And we can QC those. That works to some degree, but of course that requires sort of, if you think about it, the first transfers of malaria strip tests happened about 15 years ago, 20 years ago. You have to think very long ahead because really the uptake on those diagnostics was in the last 5 years. So at that point, all the original projects, all those original agreements were all expired. So at this point, we don't have a lot of control over anymore over what they do. But so we are really, at this point, we're looking at it similar to, I don't know how many of you are familiar with FIND. FIND has done a study on malaria test strips across the board, has found a lot of problems with them, but also some good ones. I think that's probably a reasonable approach. There is something called the WHO pre-qualification scheme that companies can submit their products to, and the ones that are accepted by WHO would then be endorsed or could be endorsed and then will be preferentially purchased by governments and by multinational donor organizations. The reality, of course, in all these things is this is all very much funding dependent. So WHO is about 5 years behind right now. They have about 30 things in the pipeline, and there's very little actually being validated, unfortunately.
So very, very nice talk and great background for all the technology. You made the point that in some situations, I think you mentioned China and India, that being able to lower the cost and accessibility to diabetes, to glucose testing could make a huge difference on public health. The last time I talked to someone who manufactures glucose diagnostic strips in quantities of 1 billion and up, they were costing about 7 cents a piece for the cost of goods sold. That's pretty inexpensive, though. Maybe you could imagine that getting down to 2 or 3 cents. So what do you have to do with everything else other than the cost of the test strip itself to bring it within reach? Because apparently the test strip cost is not the limiting cost factor anymore. At least I would guess not.
I think you're right. I think the cost in India, a strip test cost in India for glucose, that one that costs, we use 6 cents, but it's same idea, costs about 60 cents on the ground in India. The same test costs about 30 to 40 cents in China, and I think in Europe also in that neighborhood. So it's actually more expensive in India than it is here, or at least in Europe. So there's some margin here. There's just some efficiencies in terms of distribution and so on that one could work on. There is, I think, some of this is systems costs in terms of who can who can distribute and sell those readers and tests efficiently enough. And I think that's where I was talking about the business model. One of the ideas that are circulating and that I'm hoping that somebody will eventually do is sort of microcredit financed sort of a diabetes care worker. It could be a little franchise where sort of diabetes would be sort of the hub for chronic diseases in general. You don't need much other than the HbA1c reader, costs about $50. The glucose reader, you have a bunch of tests. It's sort of a central procurement for at least a village or a small area. And then that person would then, as opposed to in-home testing, the model that we have here, you'd have sort of in-village testing at least, and people would have to come every day, maybe several times a day to that person.
Thanks.
I was wondering whether you thought the mobile phone would be an important component of disease management. You just referred to the systems costs. So if a glucometer could easily communicate results to cell phone, then nudging and advising, which is so important in chronic disease management, would be simplified, or whether there's any alternative really to mobile phone for that sort of low resource setting.
No, I agree. I mean, I think everybody, or most people agree on this now. The mobile phone is going to be central to any of this, even in developed countries. There's things ranging from measurement to information to epidemiology. All these things you can really do with a mobile phone. The mobile phone doesn't replace the assay itself. It can just help, add to this.
Thanks so much.
Sure.
If anyone has any other questions for Bernhard, you can find him at one of the breaks. And now I'd like to invite the panelists up to the front.